Welcome everybody. My name is Davey Jones from Environment Centre Wales at Bangor University, and today I'm going to be talking about monitoring the environment to look at antimicrobial resistance. Within this we're going to focus on quite a few things, but really the learning objectives for today are to understand the role of antimicrobial resistance and particularly the environment in the spread of AMR. Look at it from a One Health perspective, not just looking at it from a clinical perspective, but all of the other components of the system that make it up. Look at the key sources and pathways of antimicrobial resistance in the environment. Specifically, we're going to focus on the various methods and technologies that you can use to study AMR, both for field sampling, but also the data analysis and the visualization, and then the challenges that we have in terms of interpreting that data and bringing about behavior change. And then lastly, we'll look at how we can use that information to potentially come up with strategies to reduce the spread of AMR through environmental interventions. And really that is the million-dollar question, is we can monitor AMR, antimicrobial resistance and antimicrobial resistance genes in the environment, but monitoring them is one thing, and bringing about behavior change and actually controlling AMR using that information is really challenging. So in terms of suggested pre-reading, where do you start with this subject? It's a subject that is increasing in popularity over the last couple of decades, and that's been through the advent of advances in technology and data science, and just our fundamental understanding of the importance of the problem that we have that faces the world with antimicrobial resistance and its rise. So there are many books that have been published. I have a few here that I've just put on the screen behind me that allow you to sort of get into the subject in a deeper, more a deeper focused way if you want to. They're all fantastic books. To be honest, it's impossible to read everything on this subject, as we'll see in a minute. So why should we measure antimicrobial resistance in the environment? That's one of the big questions, of course. Traditionally, we've always focused in a clinical setting, but we also need to think about the fact that people contract AMR from the environment. There's whole issues with antibiotic use in farm animals and agriculture, and also just the general environment itself and how antimicrobial resistance can affect that. So from a public health perspective, we know that when people go, for example, bathing, swimming, open water swimming, they can contract infections, and many of those are AMR infections. So it's important to understand the bacteria that live in the environment and the fungi, and what sort of resistance genes they carry and whether they're on the increase or decrease. From an animal health perspective, we know that we use a lot of antibiotics within agriculture to control diseases within animals, and of course, it's the judicious use of those antibiotics that we should be doing. But we know that the antibiotics that you feed to an animal come out the back end of the animal in urine and feces, and they go into the environment, and they also give rise to not only antimicrobial resistance within the animal gut microbiome, but also within the environmental microbiome as well. So that sort of leads us on to the ecosystem health, is that just putting antibiotics into the environment, either from wastewater discharges or from animal wastes, is basically driving this antimicrobial resistance throughout the whole of the microbiome, the global microbiome. So it's not just about humans, it's about changing that resistome of the world. It's also important, I think, for identifying trends. By monitoring the environment, we can see whether antimicrobial resistance is going up or down, depending on the environmental niches that we're monitoring. And we can also identify potential hotspots where we have this evolution of antimicrobial resistance and the potential for horizontal gene transfer. So the movement of these antimicrobial resistance genes from one organism into potentially something that is more harmful. So there's some points in the environment where that's important. It's also important to monitor antimicrobial resistance to assess risk. So, for example, if we all went swimming today and you wanted to know what the risk of contracting antimicrobial resistance is from swimming in the water or playing on the beach for your children or for yourselves, how do we evaluate that? What tools do we have to come up and say, it's risky today or it's not risky? And those are the sorts of advances in the field that we're going to have a little talk about too. In terms of interventions, if we want to control antimicrobial resistance, I think it's important that we understand how it behaves in the environment, because unless we understand its persistence and its fate and its behaviour, we have no way of really designing effective mitigation strategies. So it's that sort of fundamental knowledge which is also really important. And then hopefully all of that information can be used to basically design successful interventions to reduce the levels of antimicrobial resistance. But also, if we are bringing into play interventions in clinical settings, for example, then we need to be able to sort of assess not just whether we're reducing antimicrobial resistance in hospitals, but also whether that's having a downstream effect into the environment. So just to sort of highlight the challenge that we have of information overload in this age, this is the number of studies that have been published on AMR since the year 2000. And there's about 20,000 journal papers now, and that's increasing in an exponential way. And you can see in the most recent years, 22, 23, there's a bit of a COVID legacy here. There's a bit of a slowdown in the number of publications, but you can guarantee that that's going to take off as the rise of the importance of antimicrobial resistance and the amount of people working on it. Traditionally, it's always been done mainly from a clinical perspective, but the number of studies now on the environmental side of AMR are increasing. So we're suffering from information overload to some extent, so it's important to use new tools, whether they be AI or machine learning tools, to be able to synthesize that information and come up with some of the answers that we have. But I think one thing I want to do right from the start is say it's not just a simple case of measuring. So the easiest thing probably of this to bring about changes to antimicrobial resistance in the world, the easiest thing is probably to go and collect a sample. Then the next easiest thing is to analyze it and see how much antimicrobial resistant bacteria or fungi are in there, using bioinformatics or other things. But after that, that's where the challenges really start. And you have to think about this as a core system that you have to be able to look at all of the bits of the jigsaw if we're going to solve the problem, of which the environmental surveillance is one part of that. So for a start, we have to think about the data integration, which sits in the middle of here. It's not just about understanding what's in the environment, it's understanding all of the other factors that come into play. Water flow, contact, people's movement, animal movements, all of these other data streams can be useful. And they can be brought about through what we call infovalence, taking lots of different information streams in and funneling it into the data integration. And also environmental modelling. Have a look at that as we head on into the talk. But also, if we want to look at risk, it starts to come into the play of the sort of social sciences, ethics and acceptability. Some people are quite happy with us monitoring and doing what we would call surveillance. Other people are very unhappy about that type of approach. So we need to be really careful about how we go about monitoring things and that people don't feel that they're being invaded. And also health economics is on the bottom here. In theory, we can measure everything. That is not a problem. We can go through the environment and monitor antimicrobial resistance everywhere, and it would be brilliant. However, it's going to be extortionately costly and nobody's ever going to sign up to that. So when it comes to the health economics, we need to think where best do we put our resources to maximize the delivery of the solutions. And that is a big challenge so far in the environmental monitoring space. And then it's about how we use that information to make decisions. We've seen this through the COVID-19. Should we go into a lockdown? Should we not go into a lockdown? That's the million-dollar question. There's so many other things that come into play with the social, mental health, economics, the sort of industries and other things that go on, which also impact into the decisions that we make. So it becomes much more complicated at that stage. Ultimately, once we've made a decision, we have to communicate it effectively to an audience. And they need to act on it. So you need to have behaviour change. And that is, again, one of the other challenges. So the easiest bit is actually sampling the environment and measuring it. And the hardest bit is actually convincing anybody to change their behaviour. And so, we need science and social scientists, arts and humanities all the way through this sort of environmental surveillance to be able to crack the problem. This is just an illustration of the different types of approaches that we could take to look at antimicrobial resistance. There is huge amounts of data out there on this, of which the environmental part is one of those. And you can see in the list of things, I'm not going to go through them in detail for time purposes, but there's lots of things that you do yourself, like self-reporting apps or looking for antibiotic use or water quality on the internet and things. And some of those have high degree of data accuracy. Some of them capture large proportions of the population. Some of them are really good for getting national data, or some of them are really good for regional data. And sometimes the response is you can get the data almost instantaneously and in theory make a decision almost in real time. Whereas other things you can't. So there's lots of these different data streams, all with different levels of granularity, different costs associated with them. How do we most effectively bring those together? And probably the ones I'm going to talk about today are things to do with wastewater surveillance. It's one of my pet subjects, whether it be urban wastewater surveillance to look at antimicrobial resistance, or hospital wastewater surveillance, and how we bring these other data streams together. My personal view, as you'll see here, with hospital wastewater surveillance, for example, is green, 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 green. It's a perfect way to look at the flow of antimicrobial resistance genes into the environment. We're the same with urban wastewater surveillance too, some of the best approaches. But the key thing is not to look at any of these data streams in isolation. We need to bring them all together to basically maximize the decision making. So if we had to look at the top 10 challenges for environmental monitoring of AMR, at the moment we're still in its infancy. Although we've got 20,000 papers, journal papers produced in the subject area, there are no agreed methods to how we should monitor. So that means that trying to put the bits of the jigsaw together is somewhat difficult because people use different methods and use different standards and protocols. Also, as you listen to this and I said to you, well, the next challenge you have is to go outside and sample the environment. Where would you start? How many samples would you take? Would you sample on a daily basis, a monthly basis, a yearly basis? Do we need to sample every river in the UK, for example, for environmental surveillance? Do we need to sample every park? These are the challenges that we have that we need answers to. There's also variability in the samples that we take, whether they be samples from trees, samples from soil, samples of water, animal feces. They're all different and they all need to be treated in different ways. And I think one of the other things that we often forget is that the environment is where this antimicrobial resistance started. The microbes that live in the soil, they're all fighting with each other for resources. They're releasing antibiotics all the time. So there's a natural signal out there, and we need to try and subtract the natural signal from what we would call the anthropogenic signal. So the things that we're doing to make it worse. We need to look at how to data share and integrate that data and how to interpret it and compare different studies with each other. There's also the need for more collaboration. So animal scientists working with water quality scientists, working with soil scientists, working with crop scientists, working with clinicians, working with the GP Sentinel surveillance network. Lots of these things need to go together under the One Health banner. We can't do it alone because all of these things connect to each other now. And also, one of the key challenges is getting enough resources and money to be able to do it effectively. And I think, as I've said earlier, one of the biggest challenges I feel is how do we communicate this effectively with the stakeholders? If you talk to them about antimicrobial resistance, they say, well, is it going up or down? And you say, well, it's different for different genes. They say, well, haven't you just got a single number? Can't we just put a metric on this and, say whether it's going up or down? And it's not as easy as that because there's so many different genes, as we'll see in a minute. And then ultimately, bringing about actionable change is the problem. So getting into the core of the subject, there is really two main approaches to sampling antimicrobial resistance in the environment, or sampling what we would call the resistome, the full array, the full suite of genes that exist. So we can use quantitative PCR, so targeting specific genes that cause antimicrobial resistance, or we can take the metagenomic approach where we sample all of the genetic material or a big proportion of it within the microbiome, and we find out how the diversity of genes that are there, and if we do deep enough sequencing, we can find out which organisms contain those genes. So for quantitative PCR, this is the approach that we use in our lab mainly because it's the cheaper approach. It's got really high sensitivity. It's highly quantitative, so it can tell you whether there's 10, 50, 1,000, 1 million copies of an antimicrobial resistance gene in the environment. How you translate that into risk is a different question, but it is high throughput, and you can do this en masse now. You can run hundreds of samples in a day. The downside of it is that sometimes the number of gene targets that you can do, so there may be 800 antimicrobial resistance genes that you might want to measure, most of them won't be there, so you might just target the most important ones, which means that you miss the new ones coming into the system. And it gives you no idea about which organisms have the genes. So you don't know whether it's in a pathogen or it's in a nice friendly bacteria. And that obviously has an impact on the outcome clinically. So the metagenomic approach, which is probably the direction of travel for the field at the moment, is a more untargeted approach. So you get all of the genetic material out of your sample, you sequence it all, and you can discover lots of novel genes. Most of the genes in there, of course, you're not interested in. They're just metabolic genes and things. But if you go specifically searching for all the antimicrobial resistance genes, you can find lots of them. And if you do deep enough sequencing, so using what we would call an Illumina NovaSeq, for example, you can then ascribe different genes to different organisms. So you can see which pathogens you might be interested in. Staphylococcus, for example, which genes does it have, for example, so which you can't get from quantitative PCR. The downside is that it's expensive, but computationally, you need lots of bioinformatics and supercomputers to be able to process the information. Ultimately, that will be the direction of travel, I think. Here's just one graph. This is one illustration of the way. I'm going to talk about lots of different ways that you can, display data, because there's lots of different challenges here about how to process this data. On the top, you've got the Takara high throughput PCR system, which will analyze 96 antimicrobial resistance genes at a single time. And then you've got the Illumina NovaSeq at the bottom, which analyzes thousands of genes all simultaneously. If you compare them against each other, they actually give very similar results. Here we've got all the antimicrobial resistance genes down the y-axis, and we've got paired samples here from different wastewater samples collected over time. So this is human wastewater collected from a community, and we're looking at the antimicrobial resistance genes that are in there using the two approaches. And the size of the bubbles essentially dictates how many there are there, so the relative abundance. And you can see that the green one is the metagenomics, or the sort of light blue color, and pink is Resistomap, which is the commercial operation that does the high throughput qPCR. Generally speaking, they're very similar, but there are some differences between them. So both approaches are good. As I said, the Resistomap is cheaper. The metagenomic gives you more information and more depth. But either approach is applicable really for doing this. But this is just one idea. You can see all the diversity of genes that we're looking at here. We're never going to get a single number antimicrobial resistance in the environment because there's so many, and some are more abundant than others. So which are the important ones is the million-dollar question sometimes. So I've got a number of case studies that we're going to look at to exemplify the use of environmental tracking of AMR, just to give you an idea about the flavour of the sorts of things that we can do. There's so many things that you can do, it's impossible to do it all today, but hopefully we'll give you some ideas. So this one's about wastewater, human-derived wastewater. I like to call it citizen science because we all contribute to it. Every single day we go to the toilet, we basically flush the contents away, what we used to call flush and forget, or dilute and disperse. But within that rich tapestry of wastewater that we've got in our bottle here contains all of the sort of signs of our gut microbiome, all of the antimicrobial resistance genes that we're carrying within a population. And also it contains all of the antibiotics that we use. So we can measure not only the antibiotic use within a community, but also the resistance genes that we have. But we're not targeting individuals. Nobody in this room, I could say, you have this AMR profile and you're using these antibiotics. That's not the way it works. We're analysing lots of people in the community. So yeah, everybody contributes unless you have your own septic tank, which we don't monitor. So classically, this type of approach, wastewater-based tracking, so going to a wastewater treatment plant, the sewage plant, collecting a sample, putting it in a bottle, taking it back to the lab, spinning down the solids, extracting the resistance genes and then analysing them using qPCR or metagenomics. You can capture 100 people connected to a wastewater treatment plant or 1 million. So, for example, London captures 1 million at the Beckton wastewater treatment plant. If you go to a small village in Wales, you collect 100 people. Generally, we don't go less than 100 because of the ethics of potentially identifying individuals at that stage. It's cheap, it's simple, and it captures everybody nobody is missing out in this surveillance system. So everybody is captured. And that's important because in COVID, for example, certain sectors of the population refuse to get tested or are reluctant to get tested. In this situation, we capture everybody. So ethically, people actually quite like wastewater testing. They're not against it compared to other surveillance approaches. So it's publicly acceptable, easy to interpret the data, and we can measure antibiotics at the same time, and we can do it anywhere. So these are all of the places that we've been and tested wastewater: healthcare facilities, hospitals, care homes, abattoirs for animal resistance, airports. We've done Heathrow, for example, quarantining hotels to look at people coming into the country to see what AMR is crossing transnational boundaries, halls of residence at the universities, suburbs, ferries, travel hubs, you name it, we've been there and sampled their sewage. Some people might not think it's glamorous, but the science is real. So this is the system here, and we can do community-based AMR monitoring. So that's where we go to the wastewater treatment plant, which is connected to the community, so that everybody who goes to the toilet during the day, basically their sewage travels to the wastewater treatment plant, and we have an autosample there and we go and pick it up. But we can also do it the same thing at the hospital. So if we have an autosample at the hospital taking their wastewater, we can see which antimicrobial resistance genes are on the rise, and hopefully that provides an early warning system so that they can put interventions in place before it gets too bad. Similarly, you can also use this wastewater to track things like norovirus for the hospital so that we can prevent ward closures. So it's like multifaceted the way it works. But similarly, the wastewater discharges into the environment. So we think that we have, wastewater treatment plants completely remove all of the pathogens. They don't. They're about 99% efficient. In addition, we know that when it's high rainfall, we get raw sewage being discharged into the environment containing all of our ARGs, the antimicrobial resistance genes. So they can end up in the rivers, or they can end up going out to the ocean where people are bathing. And the biosolids, so the solid fraction from the wastewater treatment plant, goes onto agricultural land. So we've got a risk pathway there as well. So that's part of our environmental monitoring system. So if we put autosamplers at the hospital, going into the wastewater treatment plant to tell us about the community, and then on the outflow from the wastewater treatment plant to tell us what's going into the environment, it provides a really good surveillance system. There's lots of ways to capture samples, and these are some of the different approaches. So we've got some autosamplers at the top. They're refrigerated. They sit at the wastewater treatment plant and they collect samples every 20 minutes as a composite, and then somebody comes along in the morning, takes it away to the lab and analyzes it. We also have people fishing in manholes. So you have a bucket on the end of a chain or something that you lower down and you can tackle suburbs. So this has been really pioneered, this sort of suburban sampling to see which parts of a community are more at risk of carrying antimicrobial resistance genes. We've got Dan here in the top corner, where I like to call it pond dipping, where he actually has bits of cotton swab on the end of a bit of string, or other materials, electronegative filters, and you can put those into the wastewater treatment plant flow lines or in sewers, and then you can leave them in there for 24 hours, pull them out, and it captures all the bacteria on there, and you can again send it away to the lab to be analysed. Poor old Dan. We've got the autosamplers outside hospitals, so you can deploy them wherever you want, really, outside a care home or something. Again, this is one in the field sampling, and to the bottom one you can see John there in his PPE equipment with one of our refrigerated autosamplers. And then in the environment, you can just go and pick them up and put them in tubes, sediment, for example, and bring them back. So I've got a few examples of how we might use this information and the sorts of data that's interpreted. And this is one of my great examples, I think, of the use of wastewater-based surveillance. So we've got a hospital in North Wales, and we've got a wastewater treatment plant in North Wales that it's connected to that takes about 15 minutes from the sewage to get from the hospital, containing huge amounts of antimicrobial resistance genes, to the wastewater treatment plant. And then that wastewater is discharged into the Menai Straits there, which has got shell fisheries, where we're harvesting oysters and mussels and feeding them to people. So there's a risk pathway there, and there's people swimming, and there's people doing surfing further up the coast and things. So this is a classic example of the flow, the One Health agenda for looking at using wastewater, which is one of our main waste disposal pathways, to look for how it spreads in the environment. And there's one figure here. Take the one on the panel A on the left-hand side. You can see the total amount of antimicrobial resistance genes that are there in the hospital, effluent, in the wastewater treatment plant inlet, so what comes into the plant, and then when it comes out and it's treated. So you can see that that treatment process is not removing all of the genes. There's still quite a large quantity moving out into the environment. And if you look at panel B here, the other graph, you can see the Shannon index, which shows that in the hospital wastewater, the diversity of those ARGs is huge compared to what's coming out of the wastewater treatment plant. So under storm flow conditions, when the hospital wastewater is discharging directly to the environment without any treatment, that is a major risk pathway. If it goes through the wastewater treatment plant and is treated effectively, it massively reduces the risk. Now we're understanding the true risks of discharging raw sewage into the environment. Obviously, it's a big topical issue politically at the moment, but it's not without risk. So that's the total amount of ARGs. But when we get down to it, we actually can see there's a lot more information in there that's richer, that looks at individual genes and cells or gene families which are resistant to certain antibiotic groups. And that's what we're looking at here. We've got 3 hospitals and 3 wastewater treatment plants that they're connected to. You can see if you look at the real one in the middle, for example, that's probably the easiest one to look at. You can see that the wastewater treatment plant profile for ARGs, so different dates, each of those bars represents a different date, is very different from that at the hospital. So the hospital is basically one of those, what we would call antimicrobial resistance hotspot. It's just generating lots of those bugs and we're discharging them into the environment. How do you translate that and use that information when you're dealing with clinical infection teams in a hospital? They would understand this. Policymakers would say, well, they're nice colours, but what does it tell me? How do I action that data? So that's one thing. And then this is the next one I think is really interesting, is when you take the metagenomic approach. And again, a beautiful diagram could hang in the National Portrait Gallery or whatever, or the Tate Modern. What does that tell us? That tells us that we've got different bacteria here, some of which are pathogens and some of which are not. And then we've analysed all the antimicrobial resistance genes, which are in green, and we can see which organisms are carrying which genes. Now we're at the level of detail using metagenomics where we can actually really evaluate the risk. So we can now look at which pathogens, for example, Enterococcus, Mycobacterium and Streptococcus here, are very connected to lots of antimicrobial resistance genes. That's not what we want. We want them to be disconnected, really. But this is telling us now which organisms we've got there and what the state of the problem is and how they're connected. We can also use this hierarchical clustering analysis, which is another beautiful picture here of the tiles on the right-hand side. And this is actually wastewater from Heathrow, looking at the planes that are coming into the airport, sampling the wastewater from the planes. We know that antimicrobial resistance is a global issue, but it's also about the movement of these things around the world. We can now evaluate which antimicrobial resistance genes are coming into the country by taking the big aircraft that have done from Brazil or wherever it is that we can analyse for dengue or whatever viruses and things, Zika and things. But at the same time, we can look at the flow of antimicrobial resistance genes. And this one tells us, here we've got Heathrow Airport, it's also got Bristol Airport on it as well. You can look at the genes of the people who are in the terminal, so that on the outgoings and versus the people that are inbound. Again, we can see the clustering. I'm not going to go into it, it takes a bit too long, but generally speaking, the general messages are that there's different communities here of antimicrobial resistant organisms coming into the country all the time. And this is part of this environmental surveillance program, is to look at not only what's happening within the UK, but also the rate of entry of these novel genes and pathogens into the country. So it's a great way to do it. You can then use that information and look at not just the antimicrobial resistance genes, but once we've got our wastewater sample, we might as well analyse loads of other things in there at the same time, the chemistry and the heavy metals and things that might drive resistance. So we can look at those and we can decide which factors are key in being associated with and potentially driving antimicrobial resistance. This is one of these scatter plots, again for the Heathrow and Bristol terminals, showing that the amount of ammonia in this case seems to be correlated with a certain driving the certain levels of antimicrobial resistance genes. Case study 2 is about cows and sheep and poultry, and also crops. So it's about understanding, the levels of antimicrobial resistance and the impact of agriculture. So agriculture is big in terms of the use of antibiotics, but soil also contains thousands of organisms that naturally contain these genes. And it's an area where we have horizontal gene transfer, antimicrobial resistance genes passing from one organism to another. And the soil resistome, most people don't really consider the soil very often, but it's the most diverse microbial community on Earth and the most abundant in terms of the amount of organisms that it contains. And it relates to the antimicrobial resistance profile relates to the level of antibiotics we use in livestock, plus pesticides that we use for fungicides in cropping, for example, and the wastes that we add to land. And we can see that agriculture is now a hotspot for the carrying capacity of antimicrobial resistance genes. So we've got 3 antimicrobial categories here. We've got chloramphenicol, erythromycin and vancomycin, and these are done on culturable bacteria, so not using metagenomics or qPCR, but just literally plating out soil organisms and seeing what they're resistant to. And you can see in the agricultural setting, the percentage of antimicrobial resistant bacteria is much higher than what we would call a semi-natural, so that's like a more non-agricultural environment, but still in the countryside, versus urban, which is higher as well. So we can see that agriculture is definitely fueling this process of the spread of AMR. And the agriculture itself is getting worse, I think, in that sense. So why do we measure soil? Soil is good because it captures all of the processes that are happening on land, and allows you to look at the diversity and the abundance and the movement of these genes, and to look at the risk of adding waste to land. And also, in some cases, it does provide an early warning system. But one thing we do need to do is link it to some other national inventories that we have. And currently, we have no national monitoring scheme for AMR in soil, something we need to do. Case study 3 is very similar, really, in many respects. Instead of using wastewater derived from humans, it's using wastewater derived from birds. So in this case, we've got birds living on a lake. They're defecating into the water all the time, or they're releasing saliva-type material into the water containing pathogens. Whether they be looking for avian bird flu or AMR, you can use the same approach. So birds are really good because they're moving between countries all the time. They're moving between regions all the time, and with them they're carrying AMR. But we don't really know what importance that has in environmental spread at the moment. So one thing we can do is you can go and capture a bird and swab it and see what profile it's got, which is ethically challenging, as well as costly, and it takes a long time to capture a representative amount of birds. Or you can just go to the pond, collect a sample of water and analyse what's in it, and it tells you. It's also important, I think, for passing off from wild bird populations into poultry populations, etc. So this is Monipa and Dennis here, 2 people from the lab, going out, collecting a sample from a bird pond, and then analysing it in the lab. Generally, it's non-invasive, so we don't have any risks of ethics or other things, and it does capture a larger population, and you can link it directly to water quality, which is good. So again, I'm just putting up some examples of the types of information that you get through taking this approach. So here we have different antimicrobial resistance gene classes to the different antibiotics, which you can see in the legend there, and you can see these are different pond samples from different parts of the UK. And you can see that each pond or each bird wildfowl lake has a different antimicrobial resistance profile, although there are similarities between them. But what's important is that in some ponds and some lakes, there's more, and in some cases there's less. In some cases there's different ones and there's more abundance. You can see, for example, the phenicol one in the yellow in the middle there is a hot spot for that. It's not prevalent in other places. So you can start to look at the potential causes of why this is arising in these particular places, and also the types of genes that are there. And so you can then drill down to the next level and have a look at all of the individual genes and see what is going on, what's driving that process. So again, it's going to another level of detail. Again, we can take the same approach and actually not just look at the genes, but who's actually got the genes and which organisms are likely to be transferring. So it's extra levels of detail. the key challenge is how do we then resolve that data to produce something meaningful for decision making. Case study 4, just to quickly move on, we can also measure rivers. It's another classic place to look at antimicrobial resistance. It's important because rivers transport things from long distances. It also takes it from agricultural land right to the coast, so it's moving things around. And with more people doing open water swimming and surfing around the UK, there's a greater possibility of people contracting these resistance organisms through drinking water during those activities, or getting infections in their, for example, in their ears or nose or skin. And also people will continue to eat shellfish, which again sucks up a lot of these bacteria. So one thing that we've been looking at is on these sewage outfalls. There's a picture at the bottom. So sometimes the sewage is discharged into rivers, sometimes it's discharged into the sea, normally from these pipes that you can't see. But if you take a boat out, you can sample around those pipes and you can see in the sediment or the water, S or W. So the sediment around there contains a huge diversity of antimicrobial resistance genes. The water doesn't, so the sediment is actually acting as the reservoir here. It's basically holding on to all of those bacteria. And so we now think that sediments are a better way to monitor the environment than monitoring the water itself. And there's a number of people around the UK who have shown similar findings. So, but again, what you can see is just the great diversity of these genes that are contained around sewage outfalls, and that does pose a risk. So we think we've treated the water, but there's still lots being released and it's still contaminating the environment. And then take it one step more back into the food chain. So now we've consumed food, we've put it into our guts, we've taken it to the wastewater treatment plant, we've discharged it to the environment, and the filter feeders in the water, the oysters and the mussels, are now taking up the fecal material out of the water that we've discharged, and now we're reconsuming it and putting it back into our cells. So it's closing the cycle completely of human effluent coming back into the consumption food chain. So, and if you eat a live oyster, it's not cooked, obviously, and basically you're just taking in all of the pathogens that it's accumulated as it's been sitting in the bottom of the ocean. So mussels are cooked, so they're slightly better, but oysters are not. So here we have oysters that have got no pollution, so that's the natural antimicrobial resistance gene profile on the left-hand side. And then on the polluted side, we can see that if you put it in polluted water where there's wastewater discharges, it accumulates new antimicrobial resistance genes and more of them. And it's the same in mussels in not polluted water or polluted water. So the next time that you go to the fishmongers and you order oysters, maybe it's time to think again. So, or maybe it's time to solve the sewage crisis, either way. And then one of the other approaches that we can take is not just to use, look at these beautiful amounts of, graphs that say the amounts of antimicrobial resistance genes, but put that into a mathematical model. And so the model that I've got behind me now shows the release of antimicrobial resistance genes. It's just one of them here, but there's lots of different ones on the far side there. And each of these antimicrobial resistance genes, we can now predict where they go with hydrodynamic modelling. And we can superimpose on here bathing water beaches, shellfisheries and things. And we can see if there's a wastewater discharge point, which is the white triangle upstream, that's where the discharge is coming from. You can see that the antimicrobial resistant bacteria and the organisms, they're not flushing out to sea and diluting and dispersing. They're flushing in and out of the estuary day after day after day, accumulating into those mussels and oysters as they filter, feed and strip out the water. And that's why we have such a big problem. So we used to think it was dilute and disperse. In fact, that's not how it works. So, this new sort of level of hydrodynamic modelling allows us to predict risk with so much more greater accuracy. What we need to do now is link this up to sort of Met Office models and river flow models so we can do it in quasi-real time. And then one of the more quirky ones that's been around more recently is to look at bioaerosols. You can capture bioaerosols with a filter device that sucks in the air, and then you can take it to the lab and measure what's in it. But that only allows you to go out, maybe you'll only go out once every month or something and do that. But you can use trees as a sort of a capture device. So if you look outside the room, there's lots of trees there. If you look at them on the surfaces of the leaves, they've got hairs on them or they've got sticky sap on them. that attracts antimicrobial resistance genes, or they stick to the surface from wastewater treatment plants, for example, which is the picture there from Bangor. And then we've got Paul George here from the University of Laval when he was visiting, taking samples of trees. And then when we look at them, we can look at the air, how many antimicrobial resistance genes exist in the air. So every day we're all exposed to them 24/7. We can't get away from them unless you're wearing a mask all the time. So they're out there, but they're in low densities. If you look on the phylosphere, which is the scientific term for the leaf surface, you can see there's many more there. And the wastewater's obviously got a huge richness of antimicrobial resistance genes. But at that wastewater treatment plant where they have these aeration systems, so to make the wastewater degrade faster or to treat it better, you aerate it. So as you're doing the aeration process, you're just releasing lots of bioaerosols to the atmosphere, which has the potential to contaminate the wider environment. And you can use the trees to be able to monitor how far that spreads. And there's a paper that's just come out from Paul George at the bottom there, which you can look at. So looking to the future, there's several emerging themes. One is that we need to improve detection of antimicrobial resistance genes using, for example, capitalizing on the use of nanomaterials for the rapid detection of ARGs, using CRISPR-Cas technologies, lots of potential there. We're going to move, I think, from having everything monitored in the lab to being it monitored in the field, so we can almost do it with your mobile phone, similar to a lateral flow device, just going out, monitoring the environment. You can look at the risk almost instantaneously. Of course, when you've got so many antimicrobial resistance genes, that's where it becomes more of a challenge. Which ones do you target? And we should start looking not just at the metagenomes, but the metatranscriptomes to see which organisms are actually expressing these genes at any one time. We're going to have better models like the one I've just put up that capture the flow of these organisms from the atmosphere, the land, the urban environment, rivers and estuaries and coasts. They're all connected by water mainly, and by air. We can have models that link those together. At the moment that doesn't exist. And that will require more machine learning and artificial intelligence. There's no doubt that there will be more research in this area. That's obvious. It might be that we bring in phage therapy to reduce the amount of antibiotic use that we have, and we can use environmental monitoring to look at the success of that in terms of reducing the amount of ARGs in the environment, and looking at how the wider microbiome works as well, I think. In terms of controlling the spread of AMR, to me, monitoring is the easy bit. Bringing about action is the hardest bit. That's keep saying the same message, really. So it's about how we bring all these data strands together, how we bring the social scientists into this equation, joining up, not having silo mentality, and bringing about behaviour change. And then in terms of what this environmental surveillance can do, it obviously can inform AMR action plans. It can prioritise the resources, the way we spend them, the way we use them, who we're targeting, whether it be targeting farms or wastewater treatment plants. We can't target everything, so we need to do it in a resource-efficient way. We can use it to produce risk assessments like those beautiful models that predict where the contaminated water is flowing and when it's safe to go in the water or eat oysters. And hopefully we'll be able to use it to look at the effectiveness of the control measures. So just to wrap up, the key conclusions. It's not just all about clinical monitoring, of course. That's absolutely the core of the issue. But environmental monitoring is definitely a huge part of that equation that needs to be done. So what we would call the One Health agenda. There's loads of environmental compartments that can be sampled. Almost everything has its own antimicrobial resistance gene profile, and every one of them tells us a story. At the moment, we have a jigsaw that we have an idea of what the picture might be. It might be a 1,000-piece jigsaw, but we've only got 100 pieces. So how they all link together is still a challenge. We're going to rapidly advance detection technologies and ways to analyse data that will be able to makers. The people who make the decisions, we're not really actively challenging yet with the environmental data, and that needs to change. So we're going to be doing more in-field testing. So you can imagine that when you go out in your bathing water, you'll be able to test for general indicators of sewage, which give you an indication of AMR, or it might be other viruses and other things that this is going to come on stream. And we need to capitalise on the big data, on machine learning and environmental modelling to link all of these things together, we're going to have supercomputing on ways to do that more efficiently. And we need to develop a national and international AMR surveillance capability. At the moment, we're doing it in a bit of a piecemeal way, looking at different parts of the system, whether it be cows or sheep or rivers or the air or wastewater treatment plants or birds or trees or crops. They all need to be joined together in a national system which is systematic, done year after year after year after year, so we can look at change. We have no system to do that. So that's the way forward, I think. And ultimately it's about promoting the science and getting it out there to make changes. So there's some further reading here that cover a number of the aspects that I've covered in the talk. So feel free to go and visit those. As I say, there's 20,000 articles on AMR in the environment, so you're a bit spoilt for choice, but there's some good reviews here that should be able to set you on the road. Just to thank the people who did the work here. A lot of this is unpublished work from Maggie Knight, Reshma, Sylvester and Kata Farkash. And also thanks to the people who funded the work, UKHSA, Food Standards Agency under the PathSafe programme, Welsh Government, Public Health Wales and UKRI, who funded several grants in this area. We wouldn't be where we are without Welsh Water, Dŵr Cymru, who allow us to sample their wastewater whenever we like, and the population of Wales. And just to say thanks for the funding for the UKRI, for the Digital Health Hub that's sponsoring this talk. So that's me. If you want to contact me, you can do it at any time. You can just email me. And we have our website there, vracra.uk, if you want to find out more about the sort of environmental surveillance work that we do with AMR. Thank you everybody for listening. Cheers.